Okay, Bio 111 students, spring 2020. First off, I'd like to start this lecture one more time, chapter 14, part 1, for Mendel, and your book calls it Mendel and the Gene Idea. In Canvas, I had titled it Mendel and Discrete Inheritance. So to begin, I would like to share my screen with you and go through some of these PowerPoints. I know we did this on Monday and there was no audio, so I'm repeating it again with audio. I am just double checking that I'm recording. Great. Okay, so let's take a look. This first picture here, this first slide is a photograph of Mendel, and he is the third person from the right here, standing holding a sprig of fuchsia, which is a flower, obviously, to express his interest, to demonstrate his interest in plants. And I wanted to give you a little bit of background about Mendel, because I think it's important if we know a little bit about the scientists that make biology happen, makes it a little more interesting, a little more real. So he was born in 1822 in Austria, which now that part of Austria isn't Austria anymore, it's part of the Czech Republic. And that's important for two reasons. The first reason is because his native language would have been German, and so when he writes his scientific articles, he's going to write them in German, which at the time, the language of science was actually French. So he's not going to get a high readership, in part because of the language barrier. And the second reason it's important is because where that country is located, its climate is rather moderate and they have a very long growing season. So Mendel is going to choose the garden pea as his experimental model system to study inheritance, and he's going to be able to get several generations of peas in one growing season, which is going to be a big advantage to him. Now, before he became a monk, as in this picture, he actually was born in a rural area. He went to university. He studied physics, mathematics, experimental design, and botany. He studied to take the exams to be a teacher, like a grade school teacher, high school teacher. He didn't pass those exams. So after that, he decided he was going to enter the monastery. Now, to us, that might seem like an odd juxtaposition of being a religious person, a monk, and being a scientist. Please remember back to chapter 1 when we talked about Linnaeus and the introduction of the idea of natural theology, that people who were scientists believed that if they studied nature in enough detail, it would help them know the mind of God. So in this case, particularly for Mendel, it didn't seem to be any sort of an obstacle to being a scientist, also being a religious person. So enters the monastery and he begins his experimental work with peas. Now, the other important part before we talk about his exact experiments is the backdrop for the time period is that this here, what I wrote on the chalkboard here, the theory of blended inheritance. That is the going theory at the time in the mid-1800s. Mendel begins his work about 1857. He's going to work with the peas for between 3 and 5 years. And at the time, the inheritance, how traits were passed from one generation to the next, was believed to be blended. And that's not super obscure. It's pretty simple. Just means that characteristics from one parent and the other parent were blended together in the offspring, and that was it. Right? There are two major flaws with the theory of blended inheritance, and that first flaw is that if characteristics are constantly blended together, we would lose variation or variety in the offspring, meaning everything would look the same. All the dogs would look the same. All the people would look the same. All the peas would look the same. And that clearly doesn't happen. The second failure of blended inheritance is that if the traits are blended together, then a trait can't skip a generation. And people had realized, Mendel knew some farmers in the area that actually were sheep breeders. And they said, sometimes we have sheep that have curly horns, and then the next generation doesn't have curly horns, and then it reappears in the following generation. So that trait of curling horns certainly isn't completely blended away because it somehow returns in the next generation or it skips a generation. that led Mendel to say, I don't think blended inheritance is how traits are passed from generation to generation. I'm going to do some experiments and see if I can come up with another hypothesis, or I have a different hypothesis. He's going to be very successful, and we're going to end up with two laws. We call them Mendel's laws, actually, of inheritance. And they're very different from blended inheritance. He has the first law is the law of segregation, which talks about how different versions of a trait can end up, or a characteristic can end up in a gamete via meiosis. And the second law has to do with when you're studying more than one trait, how the traits assort in the gametes, or how the chromosomes assort. And you have to remember, this is before microscopes were very good, and before people had actually written out or seen all the different phases of mitosis and meiosis. So Mendel's doing this almost completely blind. Okay, now in order for us to be able to study inheritance, we need a set of vocabulary. And this is the beginning of that list. The words on the pages, all the words in bold on the pages from 270 to 276, you must know them, you must learn them, because these are the terminology, these are the words, the vocabulary that we're going to use in order to be able to complete homework problems that talk about how characteristics are passed from one generation to the next. So I'm going to start with these 5 or 6 here, and then we'll look at Mendel's experiments, and then I'm going to finish with 4 or 5 more terms that you need to know in the beginning of this chapter. So let's start with character or characteristic. The character or characteristic, I'm actually going to stop here. Let's go back to the chalkboard here. That is a particular characteristic of an organism, such as in this case, we're going to talk about flower color. That's going to be a character. Seed color, that's going to be a character or characteristic. Pod shape, that's going to be a character. Height of the plant, that's going to be a character. So eventually we're going to change this word as we move through chapter 14, 15 to 16. Character actually is going to end up referring to what we would in modern terms call either a characteristic or a gene. So a gene is a particular sequence of DNA that is going to code for the proteins to make a particular character or characteristic. In the case of the flower color in peas, there's a particular sequence of DNA that is going to code for the proteins to make a pigment, so the flowers are going to be purple, or that protein isn't made, so the flowers are going to be white. Okay? Now, the second term on our list is allele. Mendel called these elements. Your textbook mentions them as traits also. And an allele, the alleles are the versions or the variety of a characteristic or a character in a population. So, for example, our character or characteristic that we're studying with flower color, there are 2 versions, 2 variants, 2 flavors, 2 types of this allele. And those are going to be, the flowers are going to be either purple or white. So we would say there are 2 alleles or 2 versions of the character for flower color. I would remind you that in diploid organisms, which most eukaryotes are, we're going to have 2 alleles for each character, one from our mother and one from our father, or one from the male parent and one from the female parent. Next word on that list is P generation. And P stands for parent generation, and this is also called, they're referred to as true breeders. This is an organism, the parent generation is an organism that has been cultivated. In the case of the peas, Mendel is going to reproduce peas with purple flowers for 7 generations. He's going to take purple flowered peas, cross them with purple flowered peas, get the seeds, plant the seeds, see that they all have purple flowers, and then do it again for 7 times to be absolutely sure that his parent generation is pure and only has, or is true, only has the alleles for one of the versions of the allele, either purple or white in this case. Now we're going to see in his experiment, he is going to take a true breeding purple flower and cross it, that X stands for crossing or mating, with a true breeding white flowered pea plant. And the first set of offspring he is going to call the F1 generation. And the F1 generation, that stands for first filial generation. That's what the F stands for, filial. These are the babies, if you want to think of it, the progeny. We'll sometimes see that word in the problems, or the offspring of the P generation. The F1 generation is always a hybrid. It always has one allele from the purple parent and one allele from the white parent. When these F1 generations are left to self-fertilize, they produce an F2 generation, or the second filial generation. Okay, these are always the offspring of F1 crossed with itself, or an F1 crossed with another F1. So in plants, it's going to be the flowers are going to be self-fertilized. In animals, this would be like if we were talking about fruit flies, this would be maybe taking a female fly and a male fly that technically would be siblings and then crossing them, okay, to get to the F2 generation. All right, now let's take a look back at Mendel's actual experiments. And I put this picture in here, this photograph, so that you guys could all see, to remind you what the pea looks like, what a pea plant looks like, and the fact that each pea plant is going to have multiple flowers on it. Okay, so it's not just one plant with one flower, it's multiple flowers. And this panel here, figure 14.2, this we're going to walk through very simply what Mendel actually did in his experiments, right, his first set of experiments. So I would start with reminding you about the anatomy of a flower or an angiosperm, a flowering plant. So in the case of angiosperms, peas are angiosperms, notice that we have male gamete producing anthers here, or stamens, stamens here that are going to produce pollen grains. And we have the carpel here, which is where the ovule is, where there are several eggs that would be the female reproductive parts. Okay, so we have male gametophyte, female gametophyte in the same structure, and the male gametophyte is going to produce pollen, and the female gametophyte is going to produce eggs. Now, what I want you to notice is this is the P generation as it's marked here, right? So these are true breeding, or the result of 7 generations of crossing purple with purple and white with white. And now Mendel's ready to begin his experiments. And he wants to be very sure that the purple flowers aren't going to self-fertilize, or that an insect or the wind or rain isn't going to do this fertilization by accident. So what he needs to do is first he goes in and carefully opens up every purple flower on that pea plant and removes all of their stamens. On the white pea plants, he's going to take the stamens and very gently collect the pollen from those stamens, and then he's going to transfer it to the purple flower here, to the eggs of the purple flower, so that he knows that the zygotes that are going to be formed are going to be formed from purple derived eggs and from the white flower, the pollen. Okay. He is going to also complete what we call the reciprocal cross, where he would have taken the pollen from the purple flower removed all of the stamens from the white flower, and then transferred pollen from purple onto the carpel of the white flower. That reciprocal cross turns out not to be super important in his results. It only is important when the trait is actually carried on the sex chromosome, which we'll talk about in the next chapter. Then you wait until the seeds are formed. Remember, these seeds in the pea pod here, they're all going to be 2N, so they're going to be diploid. We're going to plant them in the ground, wait till they grow, and then once we see what color the flowers are, then we can write that down in our notebook. Okay, so this would be the first filial generation, and Mendel crossed true breeding purples with true breeding white flowers, and all of the F1 generation turned out to be purple. Okay. Now, to carry on, this is an abbreviated version of that experiment, right? So purple, remember that small x means that you're fertilizing or cross-fertilizing these two organisms. All the F1 generation were purple. Then he's going to allow those to self-fertilize or cross-pollinate. So what we mean by that is he wouldn't have to do all this experimenting again, this very careful cutting out the parts. All he would have to do would put maybe a small material fabric bag or square over each of the flowers and then tie it with a string. You could use cheesecloth, a couple layers of cheesecloth, or maybe muslin or linen. And then you would give the stalk of the plant a shake, and now all of these flowers would fertilize themselves, meaning the pollen from inside this flower would fertilize the ovule inside this flower. So it would be a self-fertilization. That would end up with, then we would wait till we get seeds, plant the seeds, wait for them to grow, and then we would look at the flower color of the plants. And Mendel was quite surprised because in this F2 generation, most of the plants had purple flowers, but there were a small proportion of the plants that did produce white flowers. So remember, the blending inheritance would say that isn't possible. Once you have purple and they're blended together, you would never see the white flower again. Obviously, that's not true. This leads Mendel to his first law of principle of segregation, or law of segregation. And the other thing that Mendel noticed was the proportion of how many purple flowered plants compared to how many white flowered plants. Remember, he studied physics and math, and so he also wondered if this was universal for all the traits in pea plants or just for the trait that he had studied, flower color, just the characteristic of flower color. So he conducted experiments, 7 different sets of experiments, where he first bred true breeding or pea generation for the different alleles, the different versions of the trait, and then cross them F1, parent with parent, and then let the F1 self-fertilize, and then he counted the results of his F2 generations. And I do want you to take a minute to think about this, because look how many seeds he counted. Thousands of seeds, hundreds of plants. And the mathematician in him saw that the percentage or the amount, the fraction of one of the alleles to the other one was approximately 3 to 1 every time. This isn't going to be perfect because it's an experiment, and he was doing it out in the wild, out in the environment. Maybe he had rows of peas, but he couldn't prevent every single flower from never ever getting fertilized by another mechanism than his careful dissection. Now, there are a few more words that we need to cover before, and terminology, vocabulary that you have to know before we can move on. In the next video, we'll talk about how we can take these Mendel's results and draw them inside of a Punnett square. So I'm going to go back to the chalkboard here real quick. And we're going to talk about a few other words. Phenotype. Phenotype relates to the physical appearance of the organism. pH, physical, pH, phenotype. That may help you remember what the phenotype is. People will also say this is what the organism looks like. This is what you see, what characteristic you see. And then Mendel also discussed his elements or alleles in terms of the dominant allele and the recessive allele. And I want you to be careful not to think of the dominant allele as the stronger allele. That isn't what it means. The dominant allele is the allele that is seen in the phenotype of the F1 generation. And I realize that's a little repetitive, the allele that's seen in the phenotype, because I know you just learned the word phenotype, so I'm trying to use it in the context of the dominant allele. Think about our pea plants that Mendel was breeding, right? He crossed purple flowered plants with white flowered plants. All the F1 were purple. So in the case of the characteristic or character of flower color in peas, the dominant allele is purple. The recessive allele, that's the allele for a character, right? Let's have room to write that, for a character that is masked or hidden in the F1 generation, phenotype. But, and this is an important but reappears in the F2 generation. So we don't see it in the F1 generation. All the pea plants were purple, had purple flowers, but it reappears in the F2 generation, and usually also in a smaller proportion. This would be the 1/4 proportion in a monohybrid cross. We're just looking at a single trait. So in the case of our flowering peas, the recessive allele is going to be the white, the allele for white. And we have one more word to do, genotype. No, sorry. We have more than that. We have a few more words I need to erase. This word, homozygous. So try to remember, always, always, always be thinking about the organisms that we're discussing are diploid. They are two-end. They're eukaryotic organisms, pea plants, plants in general. Homozygous relates to describing the which alleles an organism has and how many copies of identical alleles they have. So in the case of homozygous, this means homo for same. The organism has 2 copies of the same allele. So 2 copies of the allele for purple or 2 copies of the allele for white. Pea generation and true breeders, because in the problems they're going to use those words sort of interchangeably, are always homozygous, right? So these guys, pea generation and true breeders, anytime you see that word, the alleles are going to be homozygous. Heterozygous, right? It's going to be the combination, the opposite of this. The organism is going to have 2 different alleles for a character or characteristic. The F1 generation, right, always heterozygous. 1 allele from purple parent in our example, and 1 allele from the white flowered parent. So the F1 is going to have 1 copy of each of the color versions, right? The character for flower color, because purple is the dominant allele, that's what we see in the phenotype. This is the last word, I promise. Genotype The genotype is defined as just the alleles an organism has that are present, you could say, that are present in the organism. And the choice is going to be, the genotype is either, we would describe it as either heterozygous, one of each of the alleles, or the genotype homozygous, two alleles of the same thing. Okay, and we'll use these words when we start to talk about our Punnett squares and when we actually try to solve the genetics problem.